Hi, my name is Jack Hoover, and I'm going to be presenting a paper titled Extracellular Matrix Stiffness Determines DNA Repair Efficiency and Cellular Sensitivity to Genotoxic Agents. So it's first important to understand some background regarding DNA double-strand breaks and the way that cells typically repair these. So what we should look at here are first the recruitment of these phosphorylated proteins, starting with MDC1, followed by RNF8, and then these mechanisms and complexes that regulate the ubiquitin conjugation. So there's formations of long chains of ubiquitin, which is a different protein. And as we can see here, it's used in several steps of the process, these ubiquitin chains, to regulate the binding of the proper proteins to the correct places on the chromosome. And so ultimately, this will lead to the recruitment of crucial DNA repair proteins such as BRCA1 and 53BP1. And so the names of these proteins will come up again in different results of the study, and we'll kind of go over again these steps in the process, but just a brief overview is necessary. So we now want to look at the significance of the article's research question, which is how does ECM stiffness influence sensitivity to genotoxic agents? And so of course we know that signaling from the ECM is crucial in all sorts of cell functions, such as proliferation, cell death, and differentiation. But it's also important to understand that ECM stiffness in particular not only influences these functions, but it can also be the cause of aberrant cell behavior. So for example, low stiffness substrates have been shown to activate tumor suppressor proteins in previous studies. And so this study is actually looking at a previously unstudied phenomenon, which is ECM stiffness influencing sensitivity to genotoxic agents, which has sort of similar implications in terms of potential cancer treatments and other therapeutics by exposing how this mechanism works and some of the molecules involved. So the first finding of this study was that low substrate stiffness impairs double-strand break repair. And so initially, with data that is not shown here, they found that tumors with greater stiffness were actually less responsive to chemotherapy than tumors with lower stiffness, and that stiffness was measured by the amount of certain ECM components in the tumors. And so this prompted the researchers to conduct these experiments in vitro in which they cultured human embryonic kidney cells on substrates with varying Young moduluses, ranging from 0.5 kilopascals all the way to 30 kilopascals in glass, so varying stiffnesses. And what they found was that after treating with certain genotoxic agents seen in panels D through G, like radiation in panel D, as well as the drugs in panels E through G, that the cells cultured on less stiff substrates, so cells cultured on substrates with Young's moduluses of 0.5 kilopascals and 1 kilopascal, had significantly lower surviving percentages than those cells cultured on the much stiffer substrates. And so this indicates that cells on the less stiff substrates were more sensitive to those genotoxic agents. Also in panel H, they used a drug that's a CDK4/6 inhibitor instead of a genotoxic agent. And so we see there that the results were actually flipped in that the cells on the less stiff substrates were less sensitive to that drug. And so this indicates that the sensitivity is specific only to DNA damaging agents. They also wanted to look at if there was a delay in the double-strand break repair. And so in panel J here, we can see the measurement basically of the initial steps of the gamma H2AX foci, which is the initial step in that repair mechanism. And we expect that to increase significantly immediately following radiation or genotoxicity. And then we expect that to decrease as the repair mechanism takes place. But as you can see, the cells that were cultured on less stiff substrates actually had significantly higher levels of that for a longer time following exposure to radiation. So this indicates that there was a significant delay actually in that repair mechanism in those cells on the softer substrates. So the researchers then wanted to understand at exactly which step of the repair mechanism this inhibition was occurring at. And so what they did was basically measured the amount of foci of each component of the mechanism in a stepwise fashion. So you can read this figure sort of as A through G in chronological order of the mechanism of double-strand break repair. And so we have the cells cultured on varying stiffnesses that were either exposed to radiation or not exposed to radiation. And we can see that in panel A here, that all of the cells on any stiffness of substrate exposed to radiation had significant higher levels of gamma H2AX, which is associated with that double-strand break repair, compared to cells that were not exposed to radiation. So this is expected. And then we actually see the same results in MDC1. So this is the next step of that repair mechanism. We see the same in RNF8, so that's the next step. So far, none of these steps have been inhibited by the soft substrate. But then when we get to FK2 in panel D, we can see that the cells on the 1 kilopascal and 0.5 kilopascal substrates had significantly lower levels of FK2. And so this step is associated with RNF8, mediating that ubiquitination, so that ubiquitin conjugation, the formation of those long chains. This is what's being inhibited here, as seen in the fact that this is the step where we initially see lower levels of FK2. And as you can see, all the steps downstream from that, so RNF168, and then the recruitment of 53BP1 and BRCA1, we see similar results where there are significantly lower levels of all of those proteins in cells that were cultured on the less stiff substrates and were exposed to radiation. And so, again, in panel H there on the left is sort of just a helpful graphic reminding us of how these steps are laid out and the exact kind of area that is being inhibited by the soft substrate. So it's that RNF8 being able to signal for ubiquitin to form those long chains that allow the binding of repair proteins. So at this point, the researchers understood that low substrate stiffness was inhibiting double-strand break repair at the step where RNF8 mediates ubiquitin conjugation. But they then wanted to further explore this mechanism by seeing exactly what proteins or kinases this is dependent on. So this actual inhibition is dependent on. And so there's a class of kinases called hippokinases involved in these pathways. Specifically, these include MST1 and 2, as well as MAP4K, 4, 6, and 7. And so what the researchers did was they engineered cell lines that had either just knocked out MST1 and 2, so that's MM2KO, or that had just knocked out MAP4K, 4, 6, and 7, so that's MM3KO. And then they also engineered a cell line called MM5KO, which knocked out both MST and MAP4K kinases. And so what they found was that, again, in this sort of chronological stepwise fashion where each panel indicates the next step of the repair pathway, that cells with MM3KO and MM5KO actually had significant higher levels of foci at FK2, 53BP1, and BRCA1. So those cells actually fared better at their DNA double-strand break repair than those with just the MM2 knockout, as well as the wild type with no knockouts, so MM0. And so this indicated that inhibition was actually dependent on the MAP4K 467 hippokinase. They saw the same results, as you can see in panels K and L, on the effects on non-homologous end joining, as well as homologous recombination. And so these are the strategies that cells typically use to repair the double-strand breaks. And so again, ultimately, this just indicates that MAP4K 467 is what this inhibition is dependent on. So this is the kinase that must be phosphorylating something that is inhibiting RNF8 from triggering that ubiquitin conjugation. And so then through several other experiments that aren't pictured here, the researchers actually found exactly what MAP4K 467 was phosphorylating. So they found the specific amino acid on ubiquitin that was being phosphorylated that was potentially inhibiting RNF8 from triggering that ubiquitin conjugation. And so what they did was they engineered two different cell lines. They engineered T66A, which basically never had phosphorylated ubiquitin. They altered the amino acid that was typically phosphorylated so that it could never be phosphorylated, even with low substrate stiffness. And then they also engineered T66E, which had ubiquitin that was engineered, that had an amino acid change to basically always appear phosphorylated, no matter what. And then they exposed these cells, they also had the wild type as a control, and they exposed these cells to radiation and then measured the foci of each protein associated with each step in the repair pathway. And what they found was that at FK2, the cells that had the T66E, so the always phosphorylated ubiquitin, had significantly lower levels, and again, downstream at 53BP1 and BRCA1 had lower levels of both of those proteins as well compared to the wild type in the T66A, so the never phosphorylated ubiquitin and the wild type. And so this showed that by phosphorylating ubiquitin, you inhibit basically the exact same step of the repair pathway. And so this is very well what could be at play here with the low substrate stiffness causing inhibition of that step as well. And so then to sort of bring all these findings together, the researchers used those engineered cells, T66A and T66E, as well as the wild types, and they cultured them on stiff substrates at 30 kPa or 1 kPa for the much less stiff substrates. And what they found, which is really interesting, was that so at FK2, again at that step, which is where we first see any differences across all substrates, we see that the T66E cells had significantly lower levels, so had inhibited double-strand break repair at for the soft substrate and the stiff substrate. So it didn't even matter if it was if the cells were cultured on a substrate that had a Young's modulus of 30 kPa or 1 kPa. If ubiquitin was phosphorylated, as it was in T66E, no matter what, that step of the repair pathway was inhibited. And then we see in T66A, in which ubiquitin was never phosphorylated, even on the not stiff substrate, we see pretty much recovery of those levels of FK2 as well as 53BP1 and BRCA1, indicating that by blocking ubiquitin phosphorylation, we can avoid those negative effects of the soft substrate, which were, of course, seen in the wild type as well, as in the previous studies. These findings also continued for non-homologous end joining and homologous recombination. Again, those two strategies used in double-strand break repair. And so basically this confirmed that it was the phosphorylation of ubiquitin at that 66th amino acid site that was responsible for inhibiting the RNF8 mediation of ubiquitin conjugation, and that this was ultimately responsible for the low substrate stiffness, inhibiting that double-strand break repair. And so in conclusion, the researchers found that double-strand break repair is at least partially dependent on ECM stiffness, and that low stiffness inhibits that repair. They found that this repair inhibition occurs where RNF8 mediated ubiquination is happening in the double-strand break repair mechanism, and that inhibition happens because MAP4K467 phosphorylates ubiquitin at 3-amine 66, so that at that specific amino acid site. And that by blocking that phosphorylation, you can actually maintain double-strand break repair mechanisms working properly, and that also that potentially, again, by lowering the stiffness of the ECM of a certain tissue, you could impair the double-strand break repair. And so this might be preferential in tumors, for example. And then as far as a critical analysis goes, this was an extremely thorough study. So the researchers actually repeated multiple of these experiments that I presented using either different assays, different cell types, or by measuring different ECM structures to confirm the findings that they found in the figures that were actually published in the main paper. So there was extensive supplementary data to support this. There was also in-depth analysis of potential confounding pathways or other options that are sort of beyond the scope of this presentation that get a bit complicated, but that really address potential confounding factors. And again, based on the data that I've presented, this really seems to be, their conclusions seem to be strongly supported. And the MAP4K467 phosphorylation also is consistent with the studies cited earlier, where low substrate stiffness activated those tumor suppressor genes that also included MAP4K pathways. So again, all of the data really supports their conclusions, and they were very thorough in their analysis. So there are sort of two main facets to the impact of the findings of this paper. The first was that the finding that lower ECM stiffness inhibits double-strand break repair and therefore increases sensitivity to genotoxic agents could be used, for example, in understanding how well a tumor might respond to chemotherapy by measuring its ECM stiffness. It could also be the site of attack for potential therapeutics in maybe lowering the ECM stiffness of tumors to increase their sensitivity to genotoxic agents. Also, the mechanism of ubiquitin phosphorylation by MAP4K kinases inhibiting DSB repair on soft substrates was previously unknown, and that by preventing this phosphorylation, you can negate those effects. Could definitely be useful in improving the survival of certain tissues that might otherwise be prone to that double-strand break repair due to soft substrate stiffness. Okay, that concludes my presentation. Thank you for listening, and I'd be happy to answer any questions that you might have.